So this is, we converted this NSME lecture series to be online, so more people can participate. And also this lecture will be recorded for the future access. And I just want to mention a little bit. During the lecture, you are welcome to ask questions. And I have set a Padlet for anybody. If you want to post your questions on the Padlet, please using that. A little bit later, I will post the link of the Padlet to the chat. So you can click on the link for asking questions. And today's speaker is Dr. Jacobsen. I want to just highlight that she has been recognized as both outstanding researcher and outstanding teacher. Okay, she just received the Excellence in Teaching Award last year, and I believe you received the Outstanding Researcher Award many years ago, right? So that's very cool. It's a great pleasure for us to learn from you, Dr. Jacobsen. So now I will stop talking and let you take over. So I will stop sharing.
All right, thank you very much for that introduction. Let me switch over here. All right, so that should be back to everyone being able to see that. And I will go ahead and get started. So thank you everyone for being here. I was very excited to get this invitation from the dean. It's always nice to be able to talk about my work and my students. And so I decided to actually make an entirely new talk for this with a new focus. So hopefully it works well and it flows. So the first thing I'd like to do, let's see if I can get the slides advancing, is to thank all of the collaborators and students and funding that apply to the pieces of research that I'm going to be talking about today. And I especially want to thank Marlene before I forget, because I'm going to be talking about our environmental studies area on campus, and Marlene has been absolutely instrumental in keeping that facility going, keeping trees alive, keeping irrigation systems running. So thank you, Marlene, as well as everyone else mentioned on that slide. And all of those funding sources. So for those of you who may or may not be familiar with my research, I'm going to do a one-slide overview before we move on. So in my lab, we do research on several different topics, and it includes Mediterranean-type ecosystem biology and fire ecology, global change biology, plant hydraulics, which is looking at how plants use water and respond to drought and water stress, and plant anatomy. So across all of those different research themes, since I've been at CSUB, so as of the end of 2024, my lab has published 88 papers, and that research has involved 44 undergraduate students and 14 graduate students. So I've been lucky to work with really good students, and we've been very active and been able to look at a lot of really interesting research questions. For this talk, I decided rather than focusing on a subject matter or one of these areas of research, to instead focus on a place. And I decided to focus on a place on campus. And so the theme for the rest of this talk is actually going to be research that students have done on campus and an on-campus facility. And hopefully it can highlight the importance and the value of having research infrastructure, research facilities available, and kind of long-term planning and thinking about what's good for students over time. So I will very briefly cover the most minimum of plant background that you need to understand the plant biology part, since it's all going to be focused on plants. So plants have two organs, a root and a shoot. That's part one. So if you can remember that, that will become important as we move through. And then the second important thing to know is that when we talk about woody plants, the wood of a plant is secondary xylem tissue. And so we often will just shorten that and say xylem. But when we're talking about xylem, we're talking about wood. And wood is both the biomechanical structure of the tree, but also the water transport tissue. So we call the mix of water and nutrients that move through the xylem sap. And so you might have heard the term sap before. But that is woody tissue, and I'm going to be focusing a lot on woody tissue because that's the anatomical and physiological focus of my research. And I'm going to be talking about just a few cell types within the tissue of xylem. So xylem is a complex tissue, which means it has several different cell types within the tissue. The most common cell types that we see in flowering plants are vessels, which are the big pipe-like cells, fibers, which are predominantly biomechanical cells for reinforcement, and parenchyma cells, which are living cells for storage of carbohydrates and some other things in the plant. And then there's a plus there because there can be other cells that are rarer or less common across species, but those three are the three main ones. All right, there's a lot of text on this slide and the next one, and I apologize. I'll walk you through the important features because this is a history of a place. I wanted to go all the way back to the start of that history. When I was first hired at CSUB, I submitted a National Science Foundation career grant, and that career grant was to look at whole plants. And so one of the main, this is quoting from that grant, one of the main ideas from that grant was that we wanted to be able to look at both how plants are functioning, especially the xylem, and how their structure changes, so their anatomy, from the root tip all the way to the shoot tip. So looking across the entire woody plant axis. And woody plants are big. There's a lot of plant body there. And it can be challenging to look at an entire body, especially woody roots that you have to excavate to be able to look at root systems. And I was fortunate that as I was coming up with this idea, after kind of recognizing that there was this gap where we weren't really looking at whole plants, this was recognized at the time in kind of this wonderful confluence of events as being a transformative idea. And of course, that's NSF transformative, where they capitalized the NSF in the middle of transformative. So right around the time that I was submitting this grant and requesting funding for it, this really timely formative paper came out. And so this paper, so the blind man and the elephant, basically called out the plant structure function field for being what they said, we are quote unquote addicted to small diameter branches. And so if you think about, a tree that's 100 feet tall and has an extensive root system, we were basically studying how that tree was functioning by looking at what a twig was doing in a distal branch. And that's not a great way to understand how whole large organisms are working. And there were, of course, some studies looking at roots, some studies looking at trunks, but there really hadn't been much of an effort to date to put it all together. And so I was very lucky that this National Science Foundation grant was funded that let us put in infrastructure and start to plan how we were going to look at whole big trees from the root tip all the way to the shoot tip. And so that started by looking at our on-campus Center for Environmental Studies. So for those of you who might not be aware, this is the softball field. You can see the Accardo Center here, the soccer field. So this fenced area is the environmental studies area on campus. It's overseen by the Center for Environmental Studies, which is one of the oldest centers on our campus. And with this NSF grant, we put in a 288 tree plot in the northwest corner of the environmental studies area. So we got funding, and what that meant is that we went out, we had to get permission to put this plot in. We had to clear the land, install an irrigation system. My students and I planted by hand all 288 trees out into this plot. So this is what it looked like, more than a decade ago now. The trees were very successful in establishing. We've been able to do a lot of research out in this plot. And of course, it's large mature trees at this point, and a great resource. And it's on campus, planted for the purpose of studying these trees, which means we can dig up whole trees and we can put sensors in the ground and we can put sensors in branches. So it's this amazing resource. For students, this means that it's on campus. They can walk to it. So for students that don't have easy access to transportation, for students who have limited time in between classes and going to a remote field site would be difficult, this has been an amazing biological resource for students to plan and execute both undergraduate and graduate projects. So what have we done in this place? And so for the rest of the talk, I'm going to focus on some examples of research that has come out of this important research infrastructure on our campus. So from my lab, from those originally planted trees, to date, we've had 13 publications that have been published based on data from those trees. 9 of those have had student co-authors, and 3 papers have had students as the first or lead author on the papers. Again, really amazing resource. And I'm going to walk through not all 13. I'm going to walk through a few examples of what that looks like for student projects. All right, so as a first project, this was a project. So Jessica, who is the lead author on this paper, was an undergraduate student. So as an undergrad, she worked on this project, helped write it up and analyze it. She had some additional undergraduate students that helped her as she was working on this project. And then we also had Dr. Pareth Delise, who was here as a Fulbright scholar, who was helping with the project. So a really nice active team. We were looking at how the trees grow. So this sensor that's on the tree measures how much the tree is expanding. So we can actually measure how new wood is formed as trees are growing over the season. So we were looking at different parts of trees and when they start to grow and when they finish growing, when vessels in new wood that's formed actually start to move water through them. And so this is just a quick snapshot of what we found for this. So we were measuring expansion in 3 different species in different parts of the trees. The really interesting finding was that branch tips actually started to grow first, and they actually started to grow new wood anywhere from 2 to 5 weeks before the rest of the tree started to grow, and before the leaves formed. So they actually start to form new wood before leaf out. We did some really interesting work with dye tracers, which is really meticulous, careful work. Jessica, who was the undergraduate lead on this, was the most amazing, meticulous, careful worker. I'll talk about where she is at the end, but spoiler alert, she went to dental school and she's now a dentist. So good for meticulous work. And this, what we just went through here with like one intro slide and then one data slide, is what I'm going to do for each of the upcoming studies that we talk about. So it's going to be always this format of one introduction and then one data slide. So for another study to talk about, again, from those trees, and this is pictures of students working to excavate root systems of trees out in the environmental studies area. So this was actually one of our first papers that we initiated out there. And so for this paper, this was a series of undergraduate and graduate students. This was actually started as a summer Revs Up project. And Revs Up was the precursor program to SHORE, which is now our summer undergraduate research experience. And so we had an amazing time working on digging all these trees up and looking at their structure. And then Martin Venturis, who's the last author on this paper, was a postdoc here at the time. So he was a postdoc working on this project. And so from this project, we were looking at both the xylem, which is the wood, but also phloem. So phloem is the inner bark, and that's the tissue that translocates assimilates, if we're using the most sciencey jargon we can. But in the normal vernacular, it's the tissue that's moving sugars from leaves where they're photosynthesizing down to the rest of the plant. And so we looked both, across the entire root system and the entire shoot system. This is a snapshot of what we found. So these are images at the same magnification. And so if you look at this, can everyone see my pointer? Is that? So if you look at this cell is the same as this cell, but about 1/10 of the size. So in the roots, the cells and their structures are much larger than we see in the shoot. And in the phloem tissue, we see the same thing. This stained blue structure is a compound sieve plate. And so this is a compound sieve plate in the shoot. And then the blue structure above that looks kind of like a segmented caterpillar is the equivalent structure in the root. And again, much, much bigger. So the amount of variation that's possible across these different cell types in different parts of the large plant body are just huge, huge levels of variation. All right, so next, I'm combining 2 studies. So I was fortunate enough to have colleagues who were really excited about being at CSU Bakersfield. They were excited about this tree plot resource and some of our other facilities on campus, like our micro CT imaging center. And so Martin, who's here in this, he's on the right of the bottom right picture, who was a postdoc here previously, went to University of Utah after he left his postdoc here. And so he came back from Utah for a summer of research. And then this other collaborator is Dr. Uwe Hake, who came down to Bakersfield from University of Alberta in Edmonton, Canada. And we had basically a nerd summer where we were able to just do a ton of work on the trees in that plot. And we got 2 papers out of that summer of work. We also had some undergraduate and graduate students who were co-authors on this second paper.
Was this one yours right here? Yeah. Oh, this one was yours? Yeah. Yeah. Okay, cool.
All right. So I think
All right,
you might have a microphone problem, but if everyone can still hear me, I can still keep going. All right. So this is just a real quick, neat image from this study. So looking at the structure of plants and looking at that wood structure and the cells within it, we were able to use our micro CT on campus, which is micro CT that was purchased with Brandon Pratt as PI with Department of Defense funding. And so this is an example of a piece of wood from one of these trees. And then from this, we can actually extract out all of the water moving vessels and look at what the structure of just those vessel cells are as they move through the wood. So we were able to really, really carefully and very neatly look at these networks of how water was moving through the wood for trees, and 2 really nice papers. And I just want to go back to this slide. This picture is hilarious. So this was the last night of the project, and we took Dr. Hake and Dr. Venturis to the Crystal Palace for the ultimate Bakersfield experience of live country music and giant steaks that they were impressed were bigger than their heads. So a very Bakersfield experience, both in the science and in the cuisine. All right. So for this next example, so this is a paper where we had kind of a lot going on. So we did a project that was done as part of a CURE, a course-based undergraduate research experience. So this was a project that was part of our introductory biology plants course. So the 3 students that are graduate student co-authors on this paper were graduate research assistants, but also the laboratory instructors for this course. And so they're the co-authors. Dr. Mustafa Inaja, who's the next author there, was here as a Fulbright scholar. So he worked on this project as a Fulbright scholar. And this was a really neat project. So what we did, plants where leaves come off of stems, those are called nodes, and different branches can have higher or lower concentrations. You can imagine, a lot of leaves might come off of one branch, and you might have another branch where over the same distance, only 1 or 2 leaves emerge. And one of our sister schools, Cal Poly Pomona, a master's student, had just published their master's thesis, and they found that the biomechanics of stems in walnut was impacted by the number of leaves. And it was a really nice CSU-based undergrad or master's student project, clearly written. So we read that as our course discussion paper for our first discussion. And from that paper, we ended up having the students work in teams of 3 or 4, and they came up with research questions following up on this idea of where there are nodes versus the distance between nodes, how might that affect plants? And then over 3 different lab sections, we had the students coordinate coming up with questions and developing a course-wide research experimental design so that all of us were collecting together all of the data to answer each team's questions. So what that ended up actually looking like is that we maybe had one team that were interested in the relationship between node number and wood density in one species. And maybe another team was interested in looking at the relationship between node number and hydraulic conductivity across species. And another team might have been interested in looking at the relationship between hydraulic conductivity and xylem density. So we had 4 species, we had about 8 different traits, and each of the student teams had some combination of traits or species that they were looking at for their specific question. By making sure that we had an experimental design overall that answered all those questions, but was this overall encompassing design, which took a lot of kind of planning and discussion and finagling. Each group helped collect data for all of the data. So every student group made all of the measurements. What that meant is that, one, when they actually pulled out the data for their specific question, they had a really robust sample size because we had collected data across 3 different labs. When the students presented their results to the other student groups, all of the students were at least a little bit familiar with the type of traits that the different student project results were about. So it made for a really interesting student presentation discussion. And then we were able to combine everything from across the entire course into this publication, where we found some really interesting patterns that supported this smaller study that had been the master's thesis down at Cal Poly Pomona. And so we basically found that when there's more nodes, so more leaves emerging from along a branch, the stem strength is weakened. So stems are weaker when there's more leaves emerging off of them. They're less stiff when they have more leaves emerging off of them, but they have increased stem wood density, which is kind of like the inverse pattern that you might predict kind of going into it. So the overall results were quite interesting. All right. We're getting almost to the end of the examples, just in case you're you're flagging. So another study, again, done out at that tree plot. So focusing on student experience at this campus resource. So products from a place. This is a paper that was a master's thesis. So this is Alex's master's thesis. He had some undergraduate and graduate students that helped assist on this. And so they're listed as co-authors. Really interesting work. So you can see him, digging here and marking, trying to figure out where he's going to take samples. So Alex was really interested in looking at how plants store and release water as they're experiencing increasing water stress. And so we call that capacitance. So he was interested in looking at roots and stems and at different places along the roots and at different places along the stem. Are some areas of the plant more geared towards water storage or water release, and how as a plant dehydrates? So an entire plant dehydrates during a drought or a period of water stress, but different areas of the plant are responding to that water stress differently. And that, of course, drives how the whole plant functions in response to water stress. So he did these very challenging moisture release curves. And so this is roots across the top, shoots across the bottom, and then different positions within each of the root and shoot. Really very nice study. Also did some micro CT work looking at actually where water was lost in the tissue using micro CT. So a very nice study. And then the last study I'm going to talk about is actually the one that is currently in prep. So it's one I'm currently working on writing. And all of the underlined names are student names. So this is a project that's involved a lot of different students, all contributing. And what we're doing for this project is we are actually expanding from vessels to some of those other cell types that I mentioned can be in xylem. So we're looking also at parenchyma, also at fibers, and then also at some of the cellular structures themselves. So things like pits that are in the walls, the cell wall of cells, and some other kind of smaller subcellular features. So this is just an example. These are wood cross sections, and these are all at the same magnification. And so the bigger kind of white bubble looking cells are the vessels. So just as a cursory look, you can see that panel F is the top of a plant, so a distal smaller branch. D is then further down the trunk of the tree. B is in like the main wide trunk. A is then as you move into the root system, into the wide older roots. C is further down the root system, and then E is at the root tip. So very noticeable differences in vessels. But you can maybe also start to see that there's also differences in the other cell types as well. So for instance, in C and A, the smaller cells that are in between the vessels are fibers, and they're much larger in the root than they are in the stem, which makes sense. Roots are very biomechanically different than stems because they're anchored and stabilized by soil, so they don't need to have the same biomechanics. The kind of black looking ribbons that are going through the wood, those are living parenchyma cells, and those are, again, storing carbohydrates and doing some other things. So we're seeing these really massive differences across all of these different features that we're looking at. And so we're trying to, get these maps. So each of these figures on the right are basically mapping from the root tip how it's changing all the way up to the shoot tip. So how are these things changing along the entire main plant axis? When we combine all of these together, I think it's really fascinating. So this is a complicated figure, but the summary is the kind of reddish color is the shoot data. The blue color is the root data. And you not only have a huge difference between kind of what roots and shoots are doing, but we also have very definite patterns of transitions that are happening from the tip of the shoot or the tip of the root moving into older tissues. So the plant, as a large plant body, is doing all sorts of different things in different parts of the plant. I think it's really fascinating as we start to see these patterns and think about the implications. One, can we take, one of these young twigs, and can we kind of make inferences about what's going on in other places? When we have a plant that's under stress during a drought, what can we infer is going to be the bottleneck? Where are the weak points? Yeah, so lots of different things to start teasing apart as we build a whole plant, whole organismal understanding of how these different parts are varying and what that means for how plants are functioning in their environment. So that's the science kind of one figure summary. On the student perspective, I've mentioned papers and studies that students have co-authored from this on-campus research plot. I think it's great that our campus highlights student successes. So this is just 6 stories that have been published on the CSUB website featuring the students that we just looked at their papers of. So these are students that had meaningful experiences here that prepared them for the next steps and success. Jessica, who's now a dentist, Alex, who's working at UC Santa Cruz, JC, who is just finishing her PhD at University of Utah, Mitchell Coleman, who did his master's here and is now working for Tejon Ranch Conservancy, Marta, who went to graduate school at UC Berkeley, and Catherine, who's actually a faculty member back here at CSUB now. So really nice to be able to work with talented, amazing students who have done amazing things because they are amazing people. So we're very lucky that we have great students, and it's great that we have resources and facilities that can support them in their educational journey. So switching gears from this, I just want to mention a few of the current student-led research projects that are going on in my lab. So there's always kind of a mix of projects, some that I'm working on, often associated with different funding projects, but then things that students come in and say, I want to work on this, can we do a project on it? So there's a group of students that have been working on tree ring analysis from cores of oak trees out at Tejon Ranch. And so Dr. Preth-Delise, who was a Fulbright scholar here, this is some students from the SHORE program, worked to go get cores, and now these cores are being analyzed. So we're working to develop a tree ring chronology, which is an indicator of the climate history through the oaks out at Tejon Ranch. We're working on looking at vessel length in leaves. So expanding not just from the root tip to the woody shoot tip, but then actually even further looking at how these patterns are continuing into leaves and then looking across leaves. This kind of blue image down here, this is actually a cross section through a leaf with the 2 little eye-looking structures and this circle. That's actually the woody, it's not woody, but the xylem tissue of the leaf. So it really changes in structure and arrangement as you move into the leaf. So looking at vessel length in the leaves of woody angiosperms. I actually am on sabbatical this coming year and following up on this project so that we can bring this to be ready to publish is the focus of my sabbatical work. And then, let's see here. There we go. I have students that are working on looking at tree height and vessel diameter, and they're looking not only at trees in the research plot, but trees all across campus. So if you see students out with increment borers, getting samples from trees, those are mine. And so we've been looking all across campus for that. And Leah and Marcos, who are both there, recently presented at the student research competition, but more importantly, they are presenting posters at the upcoming student poster competition. And so coming up Tuesday of next week from 11 a.m. to 1, students from across campus are presenting posters of their research. And so this is just a little reminder that if you have time and you are available, to please go see the student research posters and support our students who are doing really interesting, really important research. And so we can support our students. And then my last slide is a pitch for my class this coming fall. So this fall, I am teaching Biology 5710, which is an advanced plant anatomy class. So if there are students here and any of this sounded super interesting to you, we are going to be covering a lot of this in this class in the fall. So it's available for graduate students and advanced undergrads. If you're interested in learning more about this, please contact me and we can see about getting you into this class and if it's appropriate for you in your educational journey. But I think it should be a lot of fun. And the thing I've discovered from talking with my own students is the most important thing on this entire slide is that the e-text of the course textbook is available for free. So make sure that if you're interested, you contact me and we can talk about this class. So with that, I wanted to make sure that I didn't take too long or take up too much of anyone's time in the busy time of the semester. So with that, I'd love to talk about student research or plant research or anything that anyone might have questions about. And thank you everyone for being here.
Thank you so much. It's wonderful. It's really a wonderful presentation. I'm so excited to see so many great examples of the faculty student research work. That's truly amazing. Actually, I have read a lot of the stories that you show over there from the NSME newsletters, but this is great for me to tie those news stories with the research the students have done. That's fantastic. So there is a research in chat. What would you say is a hypothesis of the research in general?
So the overarching hypothesis that we are kind of always looking is that plant structure determines plant function. So can we link the anatomical features that we're seeing with the way that plants are actually performing? And so that's like my field is plant structure function. In animal biological terms, it would probably be called comparative morphology, but that's basically the overarching hypothesis for the entire discipline.
So this now is we enter the question answer session. So if you have questions, feel free to raise your hand or just use the Zoom button or type your question in chat because we have actually 44 participants. It's a bit difficult to navigate the screen to see who has raised a hand. So if you can type your questions so that everybody can see, that's great. Or you can speak up. We're very casual. And Anna, if you stop sharing, then maybe we can see more people
on the
screen. Actually, during your presentation, I do have a question about a nice picture, actually two nice pictures that's showing up, look like a cartoon picture. No, the picture in the animation like a tree. I'm wondering, are those pictures generated by AI?
Yes, there were a couple different, there were a lot of real photographs, but there were a couple AI generated images in there as well. It was actually, I had a lot of fun playing around with that because there isn't a lot of work done on entire root systems. And so AI makes terrible root systems. And so we had quite the conversation as we tried to come up with an image that actually showed a slightly realistic root system. And I actually tried feeding some images in as well. It was an AI experiment for me. I had a lot of fun.
That's great, but the image actually looks pretty cool. So there's a lot of questions. I mean, there's one more question here in the chat. do you have any future research endeavors aside from this?
So always, I have more research questions than I can answer in my lifetime, but I'll choose to answer that. Maybe not the way it was asked. So the research that I'm doing that's separate from student research projects. So my main kind of area that I've been chipping away at for a long, long time is looking at fire ecology and drought ecology of chaparral shrublands, which are kind of our native abundant shrublands that you drive through when you head south through the grapevine. So I'm working on a project right now on restoration of chaparral shrublands that have been degraded because of too much fire or drought. So that's ongoing work. For the 5710 class that's going to be offered in the fall, I've actually been corresponding and collecting samples from around the world. And I'm thinking, I haven't quite worked out all the details, but I think for that course, we're going to really dive into arborescent monocots as kind of a special topic as part of that course. So I've gotten grass tree and palm and aloe samples from across the globe sent from collaborators so that we have some examples of non-woody trees where we can look at that. So still working on that idea, but working on getting the samples. So a lot of different things still in the works.
Great. Let's see if we have other additional questions from the student first. So I have a couple of questions actually. One is related to the fire because you mentioned fire several times, and wildfire has been issue in the state of California. So I'm wondering how your research can be connected to the fire situation, maybe help to select a species that is more fire resistant.
Yeah, so I have several publications and studies over the years looking at kind of different aspects of fire and how plants respond to fire. So there's two basic ways that plants come back from fire. So one is that they are able to resprout. So they have buds underground as part of their root system or as part of specialized structures, and they can resprout after the fire from those underground structures. And the other is that the plant is killed and comes back from seeds that germinate after the fire. And so there's two kind of different problems there. So one thing that we found repeatedly is that the plants that are resprouting after fire during the phase when they're resprouting change how they make wood, how they transport water. So they change in how susceptible they are to drought. So if we get a fire followed by a drought, plants that are doing that, using that strategy seem to be particularly threatened. With plants that come back from seed, seedlings actually can be pretty tough, especially for some of the species that come back from seeds. But what we find is that those are plants that are dying predominantly, they're dying in higher percentages in droughts before fire. And if there's droughts before fire, then they haven't made seeds recently. And it's the most recent seed production that actually are the seeds that germinate after fire. So if you have a drought before the fire, then you might not have a lot of seeds in the seed bank. And so you can have limited seed regeneration. And so there's a lot of really complicated compounding stressors that impact how our native plant systems come back after fire, all of which have been conspiring of late to make for really challenging conditions. And this is why we're getting degraded natural systems. And so starting to think about restoration becomes important because we're not keeping our intact systems healthy.
Thank you. That's very helpful. There's one more question on the chat. Is there a similar difference in size of structural element between roots and shoots in non-woody plant tissues?
So thank you, Carl. There's two different ways, I think, to answer that question. So one, so we see similar difference in size of the structural elements in non-woody tissue of trees, which would be like the inner bark and the phloem. So it's non-wood tissues, but across the size of a tree. And in those non-woody parts of the tree, we see the same differences in sizes. So huge differences in size. So when you have a large tree organism, you get just these huge disparate structures across the size of that big organism. in non-woody plants, like an entire non-woody plant, which would be like a palm or an herbaceous plant, their body size is smaller, and so the level of variation isn't quite as high. And then we'll have to look, I haven't actually seen a lot of research, like looking at the top of a tall palm tree versus the bottom. I have a request into facilities that anytime they are going to be like removing palms on campus anytime soon, that I get to claim the plant carcass body. So hopefully they call me and we can actually cut up some big palm trees as well. But I don't have any palm trees planted in the research tree plot, so I don't have any that I can easily start to dismantle for study.
Great, thank you. So one more question. Expanding on your general hypothesis, can you provide a specific example of plant structure contributing to plant function?
Yeah. Thank you, Michelle. Yeah, so one example that's a little bit in the weeds, but one that we're really interested in. So when you have big vessels, you tend to also have big pits in those vessels, which are the small pores that connect one vessel to another for water movement. So you have big vessels with big pits, and that actually means that those vessels are particularly sensitive to water stress associated embolism and hydraulic failure. So you have large vessels that are then increasing susceptibility to hydraulic failure. And so smaller vessels, maybe in distal branches, are less susceptible to hydraulic failure. What's interesting in big plants is that there's a water potential gradient. And so the leaves and the distal branches will be at more negative water potentials compared to the roots, and the roots are, in wet soil, so they'll be less negative. So are the roots with larger vessels more vulnerable overall, but not more vulnerable for their hydraulic environment? That we're still looking at. That was part of Alex's thesis work. But, it gets tricky because there are these, it's not just that structures are different across the plant, but the actual physiological status of different parts of plants are also different. Now, that was probably too detailed, but it's a good question. Oh, and I see Marlene not only is keeping the watering system running for the tree plot, she's informing me that there's a palm tree potentially willing to be sacrificed out at the ESA as well, which is great.
So there's a palm tree. Okay, interesting. And actually, it's a very, very inspiring to see so many research projects on our own campus from the ESA area. As you know, I really want to make this area more engaging and sponsor more projects. What additional projects do you foresee that you can utilize that area, or do you think there's other improvement or other things we should do to make that area kind of better support the faculty student research?
So I have good answers for this. So one, there's already a lot of work being done out there. So in addition to the tree plot, Dr. McNish has some microcosms out there doing some aquatic research. We have, Dr. Pratt has a chaparral shrub garden, so we have a shrub garden out there as well. And then we have the southern forest area. I would love if we had the ability to access tree canopies easily, like a canopy walk or like a cherry picker. In my wildest dreams, we have some way to get up into trees while they're still intact, and we can get to the tops of trees. That the amount of questions that would expand for what our students could look at would be amazing. So that's my wish list, big dream.
So you want to have some mechanisms for you to get access on the top of the tree. I see. Okay,
or a canopy walk. We can just put in infrastructure. Infrastructure would also be acceptable.
Yeah. Okay. Very good. So Marlene will be taking notes. Okay. Carl has a question. Go ahead, please.
Not a question so much, just, with the idea of doing a canopy walk, if you turn it into a ropes course, you can charge admission to help pay for it.
Yeah, we can combine education with exercise. I like it.
Okay, great. And you talk about expanding your research during your sabbatical. Could you elaborate a little bit more about your plan? What research plan you have in your sabbatical?
Yeah, so for my sabbatical, I'm planning on looking at the vessel length distribution in leaves. And so we were able to actually take the students in one of my shore groups really wanted to look at leaves. And so I usually look at wood, which is a lot easier to deal with. And so we basically spent the summer figuring out how we can do silicone injections into the cut ends of leaves to fill the vessels so that we could then kind of handle the leaves a little bit more to look to see the pathway of vessels through the leaves, but then also the diameter of individual vessels. And so now that we have that protocol established,
Yeah, maybe Dr. Jacobsen's screen is frozen. Or maybe my networking is not working very well.
Yours is fine. It's Anna who's frozen.
I see. Okay. We can wait a little bit for her to come back. So I don't know if she can see us. Okay, so I guess we lost her. Hopefully she will be connecting back soon. So I'm going to stop recording at this point.